I think we'll shift straight over to our first keynote speaker, and it gives me great pleasure to welcome Jacky Goetz, who comes from University of Strasbourg, with the very exciting title, Nanoscale Tracking of Tumor Metastasis. Jacky, over to you.
Hi, John. Hi, Joanna. Thanks a lot. Hi, everyone. Thank you for coming. So I'll share my screen now. Just let me know whether you see it or not.
Yes, we can see it. It looks good.
It's all good? Okay.
Yeah.
Cool. So I'm very happy and excited to, let me just get rid of this because I don't see. Very happy and excited to show what we've been doing in the lab in the particular context of looking at how cancer evolves using animal models and especially imaging, because this is definitely one of the main motto of my team, is to try to track these events using state-of-the-art imaging approaches. And what you see here on the slide is quite a nice example of the way we look at cancer progression and tumor metastasis, which is by having some sort of windows within the animal. And this is one example within the zebrafish embryo, where we can very nicely interrogate how circulating tumor cells, here in blue, basically behave in intravascular environment of the zebrafish embryo. That's you can see here where the blood cells are labeled in green and the endothelial cells are labeled in red. And this is definitely one way, major way that we're using in the team to try to understand some of these steps, which are actually essential in driving tumor to form metastasis. So this is the team, the current team. So I won't have time to thank everyone, but I just place it here for two reasons, because I won't have time to thank everyone. We have an open position in the lab for doing microscopy on metastasis, so I encourage people that would be interested to apply. And also some of the key people which did the work that I will show you are not on this slide, but I will try to basically acknowledge their work during the talk. So as I told you, we are really very much interested in trying to understand how cancer and primary tumors actually evolve to metastasis. And why is that? It's mostly because, of course, metastasis we know causes more than 90% of the cancer-related deaths. And it's quite well established now that, unlike primary tumors, which can often be cured by local surgery or radiation, metastasis is a systemic disease. And despite recent advances in the past years, the mortality rates have either stagnated or even progressed for several cancers. And the vast majority of patients will die within 5 years of their diagnosis. So we think that it's quite clear that treating metastasis remains a big challenge, and understanding how metastasis evolves also remains a challenge. So in my lab a few years back, we decided to develop very fundamental, basic cancer science approaches to try to understand how primary tumors evolve to the secondary fossa, which are called metastasis. And we, as I told you, we're using a lot of imaging approaches into that. And we decided to start this type of work based on a few facts. One of them is that most of the key events which are at the basis of tumor metastasis remain very rare at the level of an organism. So you need to develop very sophisticated and well targeted imaging approaches to be able to track these events in vivo. And we thought at that time that there was still a big lack in resolution of some of the events that we're interested in, and this is what we tried to do. We're also interested a lot in trying to understand how external cues can actually drive metastasis formation. So they can be, of course, biological through the microenvironment, but also conveyed by mechanical forces. And I'll show you some work that we've done in that part. And a bit more recently, in the past year, we decided to explore a new area that we hadn't explored in the past, which is the fact that the blood circulation actually carries a huge amount of very active objects, which are extracellular vesicles that are secreted in high amounts by tumors. And we thought that we could use this property to actually apply our imaging technologies, but also try to potentially use them as liquid biopsy and identify new biomarkers of tumor metastasis formation. So what I'm showing you here is an example that is quite old now in the team, but that is summarizing some work that we've done. So in the context of today's talk, I think it's quite nice to emphasize this aspect, because this was developed in collaboration with the team of Yannick Schwab at the EMBL in Heidelberg, and Matja Karman in particular, and in collaboration with the team of Frank Winkler at the DKFZ in Heidelberg, where for quite some years we decided to spend a bit of time on trying to develop correlation approaches which allow us to combine the power of the intravascular imaging technology. And you can see one example here in the movie, where we can track single tumor cell in a living mouse brain with the resolution, because we thought that this particular event of circulating tumor cell arrest and extravasation, which is at the basis of tumor metastasis formation, lacked some resolution. So we developed procedures that allow us basically to scan this exact same tumor cell, but this time using volume electron microscopy approaches. And what you see here is one single stack FIPSAM, so focused ion beam scanning electron microscopy of this exact same tumor cell, but scanned using scanning electron microscopy at an isotropic resolution of 8 nanometers, which allows us to give to see and to have access to a big amount of information, and quite a big amount of information regarding subcellular events that might occur in that particular case. And the movie here ends up on a region where we can see this tumor cell that is actually trying to poke a very tiny finger between two endothelial cells. And here you will see the basal lamina of that blood capillary in the mouse brain. So this is one example, and this scheme quite nicely summarizes some of the events that were on which we basically worked a lot in the past year. So we are interested in how circulating tumor cells, once they are shed by the tumors, they would stop, arrest, establish stable adhesion with the vascular wall, get occluded in some cases. This is a project that we have right now, and then eventually would extravasate out of the blood circulation through diapedesis, or through a new mechanism that we've identified in our work, and I will show you that later. But we're also interested in how these tiny vesicles that I told you are shed in huge amounts by primary tumors and circulate freely in the blood circulation, how these tiny vesicles, which are roughly 150 nanometer in size, would actually be capable of priming pre-metastatic niches, so preparing the microenvironment for establishing successful metastasis. And this is a particular event on which we spend a lot of time in the past year, and I'll show you how we did that. So very quickly, how we do and develop this type of project, we mostly rely on cell culture approaches where we isolate extracellular vesicles, in that particular case coming from Zebrafish melanoma cell line that we were able to get from Richard White's group in New York. We characterize these tiny extracellular vesicles. They contain a huge amount of information, so because they are covered with proteins, of course, but they also contain a lot of nucleic acids that can be very efficient in priming this microenvironment. So we can characterize them using nanoparticle tracking assay or electron microscopy. We can perform mass spectrometry analysis on them to really characterize that they contain most of the markers of extracellular vesicles at that stage. And in that particular case of the Zebrafish melanoma model, we really made sure that they actually behave quite similarly to human models to really validate that there are good models for melanoma extracellular vesicles. So what we decided very quickly is to first establish the fact that these extracellular vesicles have this property of establishing pre-metastatic niches, and this can be done quite nicely in the zebrafish embryo, and it had been done a lot in the mouse system, where you basically prime your animals with an injection of extracellular vesicles which are derived from melanoma cells, which is then followed a few hours after by an intravenous injection of these circulating tumor cells to favor metastasis formation. So what happens in the case of animals which were injected with inert beads, you can see that there are tiny metastases formed in the zebrafish embryo. But when you have primed these animals with extracellular vesicles derived from the same tumor cells, you can see that we can highly potentiate this effect. One key question that was very of high interest to us at the very beginning of this program is to be able to visualize these extracellular vesicles, because again, they are very tiny objects that they contain a huge amount of information, but they are roughly 150 nanometer in size. So we developed approaches allowing us to basically fluorescently label these extracellular vesicles using a lipid dye. before injecting them in the animal. And this is something we do in the zebrafish embryo, but which we also do in the mouse system, with the idea to be able to map how basically these extracellular vesicles behave in a realistic blood circulation scenario. And this is one image that shows you what we can get as information. Again, the zebrafish embryo using a combination of two transgenic lines, allowing us to visualize both the endothelial cells and the red blood cells. And what you see here appearing in cyan here are the extracellular vesicles that have arrested in a very specific region of the zebrafish embryo, which is called the caudal plexus. And this image contains a lot of information. I will get back to that later. But what we were really interested is to see whether we could apply high-speed microscopy approaches to really characterize the blood dissemination of these extracellular vesicles. And I hope this is something that you can see here on your screen. It's again a blow up on the caudal plexus region. We are in the blood flow, and you can see all these tiny particles that are freely disseminating in this blood circulation are these fluorescently labeled extracellular vesicles. So this is the type of acquisition that we performed in the past. And this allows us to really nicely interrogate how they behave and how they stop in specific vascular regions. You might have spotted a few arrest events of these tiny extracellular vesicles. One just happened here. We quickly realized that a huge amount of extracellular vesicles tend to stop at specific region. And we wondered at the very beginning what these regions were. We went back and used another transgenic line in the zebrafish embryo. So this is roughly the same region, the same vascular region in the zebrafish embryo of coming from another transgenic line, where the macrophages have been are labeled in green here in GFP. And we actually realized that this cluster of extracellular vesicles correspond very nicely to a subpopulation of patrolling macrophages. So this is a population of macrophages that reside within the blood vessel and that are capable of engulfing a huge amount of extracellular vesicle in that particular environment. So we applied, because one of the black box in the field of extracellular vesicles is to understand how they can transfer their message to the receiving cell. We applied the correlative light and electron microscopy approach that I introduced before to that particular setting, where we can, again, using confocal microscopy, track single macrophage cells. So here, in that case, two macrophages which are within the vessel. As you can see, we still have flowing extravesicle in that case. And then by doing this correlation of confocal microscopy that you see here in the first line of this slide, and the electron microscopy, which is in that case serial transmission electron microscopy, we can actually correlate the position of the extracellular vesicles within these macrophages, but this time at nanoscale resolution. And actually, it turns out that most of the spots, fluorescent spots, they correlate with compartments which are late endosomal and lysosomal compartments, suggesting that actually they are very quickly degraded by this particular population of macrophages. And it's something we would like to now expand to endothelial cells, which engulf a huge amount of extracellular as well. Because indeed, one of the interests that we have is how these extracellular vesicles behave in relation to the endothelial cells. Because if you look carefully at the movie again, you can see that they are engulfed by patrolling macrophages, but are also arresting in specific vascular region quite heavily. And indeed, endothelial cells, which compose the vascular wall, can internalize this quite heavily. So we here in that particular case, we use another advantage of the zebrafish embryo, which is that it allows us to do high-speed imaging of the blood flow, but also to quantify, in that particular case, using particle image velocimetry analysis to quantify quite accurately what are the hemodynamic profiles in specific vascular regions. And one other advantage is that if you apply drugs such as the lidocaine, so a pharmacological approach which allows to tune the pacemaker activity of the zebrafish embryo heart, you can actually tune these blood flow profiles. And we use that particular aspect because we actually quickly realized that if you look again at this particular image, most of the extracellular vesicles that you see here again in cyan, they tend to arrest in specific vascular region which corresponds to permissive flow profiles. So if you take this big vessel that you see here on the dorsal part of the caudal plexus, which is the dorsal artery, you can see that we have very little amount of extracellular vesicles that would actually stop in contact with the endothelial cells. In comparison to these tiny vessels here, which are characterized by very low flow profiles, where there is a huge amount of extracellular vesicles that are stuck to the vascular wall. suggesting that actually these extracellular vesicles, they have to basically play some sort of tug of war between the shear forces that is at play within these blood vessels and their adhesive potential, making them susceptible to stick to the vascular wall. And it's something actually we wanted to test very quickly. I think I skipped that slide because it's probably not very informative, but we were very much interested in trying to understand whether these tiny extracellular vesicles would contain cell adhesion molecules that would favor their arrest in specific vascular regions and basically be able to counteract the shear forces that is imposed by the blood flow. And we got some piece of information by studying the mechanism and the biogenesis of these extracellular vesicles, which seems to be controlled by two GTPases, RAL-A and RAL-B, and I'll show you how we did that. Because a few years back, we actually quickly realized that in the context of breast cancer progression, and by using the 41 model, which is a biopsy breast carcinoma model, when we decrease the expression levels of RAL-A and RAL-B, again, two GTPases, we quite massively reduce the density of multivesicular bodies that you see here, which is actually the platform of secretion of these tiny extracellular vesicles intracellularly. So both the size but mostly the density of this multivesicular body seems to be directly dependent on RAL-A and RAL-B expression levels. But what was interesting is that the amount of extracellular vesicles that are then shed by these tumor cells, of course, is also strongly reduced in absence of RAL-A or in absence of RAL-B. And quickly, as a side comment, the expression levels of RAL-A and RAL-B seems to correlate quite nicely with a poor prognosis in the context of metastatic breast cancer. But then we performed mass spectrometry on the extracellular vesicles that are shed by these three different populations. And we actually were able to identify a list of 5 proteins, and one of them being CD146, which is a cell adhesion molecule classically expressed at the surface of the vascular wall. And this sparked our interest because this is actually what we were looking for to try to see how we could tune the adhesion potential of these extracellular vesicles. So this was confirmed by Western blot analysis. But briefly, in absence of RAL-A or RAL-B, the cells tend to secrete extracellular vesicles that have a reduced amount of CD146 at the surface of the extracellular vesicles. And we then moved back to imaging approaches to see whether this particular aspect would drive the arrest potential of the extracellular vesicle in vascular region. And we use an antibody-based approach where we mixed the extracellular vesicles with or without an antibody directed against CD146 and injected the extracellular vesicles in the zebrafish embryo, looking at their ability to stop in specific vascular region. This was reduced when the EVs were pre-treated with this particular antibody. And we moved to the mouse system to confirm that this was also the case in the context of arrest of extracellular vesicles in lungs using lung sections. But what we were mostly interested in, and which is using another imaging approach, which is a whole animal bioluminescence approach, is to see whether this has a functional effect. And we spend a little bit more time on this particular aspect where we use this priming scenario. So it's basically an experiment where you prime the mice with 2 rounds of injection of extracellular vesicles intravascularly. This allows to prime the metastatic organ before the subsequent injection of 41 cells in the tail vein in order to form lung metastasis. So if you look at these 2 first images, when you inject mice with PBS, then followed by a tail vein injection of tumor cells, you have lung metastases that are being formed. But this is massively increased when the same mice have been pre-injected with extracellular vesicles coming from control cells. But when these extracellular vesicles were pre-mixed with an antibody directed against CD146, we can see we can put basically reverse this effect and bring it back to the basal levels that we obtained with the PBS condition. So suggesting quite nicely that the CD146 is a key molecule at the surface of the extracellular vesicles, favoring their arrest in specific metastatic regions and favoring the pro-metastatic effect of extracellular vesicles in the context of breast cancer. I will spend a bit more time now on showing you how we are looking also at the arrest of circulating tumor cell, and this is the work of many people in the lab, including Nael Osmani and Gauthier Follin. And I think Gauthier is in the audience, so hello, Gauthier. Where we actually very quickly were interested in how these hemodynamic forces would actually be able to tune the ability that the circulating tumor cells have to stop in specific vascular region and to extravasate. We used again a lot the Zebrafish embryo system for many reasons. One of them, it has a very highly stereotyped vasculature, allowing us to basically merge a huge amount of embryos with the idea of showing up using heat mapping the favored position of arrest of the circulating tumor cells few hours after their injection in the Zebrafish embryo. And this is one of the heat maps that we obtained, showing that this particular spot here in this vascular region is a hotspot of arrest of circulating tumor cell. And it turns out that at the same time, the team of Klaus Pantel in Germany were actually performing very similar approaches, but this time in a cohort of patients having a brain metastasis. So this was a cohort of 100 patients where they mapped the favored position of metastatic foci in these patients and compared basically this 3D map to the perfusion profile of a control patient. And to make a long story short, there is a very strong correlation between the perfusion index of the specific brain region and the ability that basically these type of tumors had to form brain metastases. Again, we could show this very nicely in the Zebrafish embryo, where we can use the ability to tune the pacemaker activity of the Zebrafish embryo. So either decreasing it using lidocaine, as I showed you before, but in that particular situation, increasing it using another drug called the IBMX. We can very nicely characterize in many different regions what is the effect of this increased pacemaker activity that we impose to the Zebrafish. And we then combine this with another imaging approach, which is a stereo microscopy approaches, where we have a way to very quickly interrogate the ability that circulating tumor cells have to stop in specific vascular regions. And this movie show you, maybe look only at the two first, maybe I'll come back to this one. I cannot, oops, sorry. But you can focus on this particular region here, where you can see that most of the circulating tumor cells in a controlled situation would end up in that specific region I just showed you before. But when we have embryos here, the IBMX situation, where we have increased flow profiles, what happens is that basically most of the circulating tumor cells, they remain circulating. Because the shear forces that are imposed by the increased flow profiles actually contracts the adhesive potential of this particular circulating tumor cell, making them incapable of arresting in specific vascular region. We can interrogate this again using correlative light and electron microscopy, where we very quickly identify that circulating tumor cells establish quite stable contacts with the vascular wall, as you can see here from the electron microscopy images. And we are also interested using another type of approach to see to what extent actually circulating tumor cells are, and the amount of forces that is at play as soon as the circulating tumor cells that you see here are arrested in specific vascular region. And using optical tweezing technology, which allows us to trap red blood cells to really accurately quantify the amount of hemodynamic forces at play in specific vessels, we wanted to use this optical tweezing technology to try to detach the tumor cell from this initial point of arrest in specific blood vessels. But this result is mostly negative because the amount of forces that the optical tweezing setup that we have cannot actually reach this phenomenon of detaching the circulating tumor cell from the vascular wall, suggesting that the additional forces that these cells establish very quickly exceed the range of 200 piconewton. But we were able to use the optical tweezing technology in a microfluidic system, where we can actually also very easily tune the expression levels of some receptors at the surface of the circulating tumor cells. And I won't spend too much time on that, but just to show you that this allows us to identify that there are like a plethora of receptors which are used differently by circulating tumor cells to favor the first set of metastable adhesion that's basically allows circulating tumor cells to stop at the surface of the vascular wall, and another set of receptors, mostly integrin beta-1, that favors very stable adhesion in specific vascular regions before they actually extravasate and form metastasis. And on the last set of slides, I'll show you also how we use imaging to basically come up with new observations as to how circulating tumor cells stop and extravasate out of the blood circulation, which is a pre-requirement for forming a metastasis. And here, one observation was performed very early on using the correlative approach that we developed in the mouse brain, again, in collaboration with the team of Yannick Schwab and Frank Winkler, where we actually quickly realized when we looked at circulating tumor cells that had just arrested in specific capillaries, so very early events, and when we performed these electron microscopy images of this exact same tumor cell here, we realized that the endothelial cells, and I encourage you here to look at the red segmentation, they very quickly some tiny protrusion very close to the arrested circulating tumor cells, suggesting that the endothelial cells are actually very active as soon as the circulating tumor cells stop in specific vascular region, and suggesting that they actually very quickly engage this endothelial remodeling effect that is schematized here. This is a scenario that we can actually reproduce using a microfluidic system to which we can apply scanning electron microscopy approaches. In that set of images here, what you see are arrested tumor cells in a context where we did not apply flow, so where we did not mimic the situation of a blood vessel. So the cells, they stop on the surface of the endothelial cells with no major effect. But what happens when you had applied a flow that seems to be very permissive to this particular scenario? We quickly realized that the endothelial cells actually, they massively engulf this arrested tumor cell, which really nicely suggests that the endothelial cells are highly reactive to the presence of an arrested circulating tumor cell, and they tend to engulf the cell with the idea of expelling it out of the blood circulation. We can look at this using again time-lapse imaging in the zebrafish embryo. And we can again apply correlative light and electron microscopy to that particular situation. So this is an example where a cluster of cells was arrested in the dorsal aorta of the zebrafish embryo. And when we looked using a combination of serial transmission electron microscopy and electron tomography, we actually realized again that this cluster of circulating tumor cells were actually already engulfed by a very tiny layer of endothelial cell, which even established cell-cell contact quite nicely using that we could realize using these electron tomography approaches. And just to finish on that particular part, we actually very quickly realized that this particular event of endothelial remodeling seems to be, in our case, flow dependent. We identified a few genetic programs that were actually engaged, initiated by the flow. One of them being an angiogenic program that we try to impair using classical drugs meant to target VEGFR2, which is a key node for driving this endothelial remodeling effect. And just very quickly, when we use these drugs, so either sunitinib or cediranib, which is a bit more specific to the VEGFR2 receptor, in a situation where we try to look at the amount of cells that would eventually extravasate out of the blood circulation in the zebrafish embryo, you can see that we quite nicely can reduce the ability that these cells have to extravasate, suggesting that this endothelial remodeling can also be impaired and reducing the extravasation ability of this tumor cell. I think I'll skip that because I want to finish here. Just I hope I convince you that by using a combination of in vitro approaches, microfluidic approaches, the zebrafish embryo imaging approaches, and some functional but also imaging approaches in the mouse system allows us to really accurately look at some of the key steps driving tumor metastasis formation from an intravascular perspective. Of course, this is something that we continue now in the team. I just want to finish with two slides, which are non-scientific, but well, still they are, because I want to emphasize that we are part of a big consortium in France, which is called the Nanotumor Consortium, where one of the main actually idea and goal of this particular consortium is to again map key structural and protein elements in cancer cells. So using a combination of many different approaches, including structural biology that is performed here in Strasbourg at the IGBMC. And I want to finish also by saying that we just recently moved to a new building in Strasbourg, the CRBS, and I hope that I'll be able to invite you very soon in Strasbourg to visit this new building, for which we're putting quite some efforts in developing an imaging platform that I'm doing with Pascal Clessier within the building. But we also recently were able, with all the imaging partners in Strasbourg, to build this federation of imaging platforms in Strasbourg, and I'm using this opportunity to tell you that there will be an opening event of this new network in Strasbourg that will happen, I think, well, December 13th and 14th, which will be at this stage still virtual, but where I know that the registration are still open, and there's quite a good list of speakers also that will make this event very exciting. And I'll finish here by a few acknowledgements on some of the key collaboration for the things that we're developing. So I mentioned Yannick and Frank for developing the intraocular qualitative imaging approaches. Klaus for the work that we've performed on circulating tumor cells, and many other partners that are key, of course, in driving these projects. And I'll stop here. I hope I was in time.
Thanks, Jacky, for a wonderful presentation and wonderful, exciting view into your secret world.